Раз. It's constantly. It's constantly. Constantly. Oh, which one? Not good use there. Which one? Invitation for them. Раз не люблю ее. Как это? Ты меня извини, пожалуйста, да, вот у меня сейчас во вторник лекция в 16 часов с фармашами. У меня постоянная ссылка, да, я по ней зашла. Тут у меня 4 человека, остальные все пишут, киньте нам ссылку, почему нам ссылку не присылаете? Я пишу, это постоянная ссылка, она как всегда, а где ее взять? Вот ты знаешь, я не могу, я с этими постоянными ссылками с ума сойду. Может, мне действительно надо было копировать, прежде чем войти и отправить им, да? Да, кто-то просил, я ее кинула, не знаю, и в деканате, где она болтается, я даже сама сказать не могу. Ты помнишь, ты ее скопировал и своему старости кинул? У тебя 5103. Напиши ему, чтобы он остальным скинул, куда-нибудь, чтобы они общались. Есть. Угу, подожди-ка. Нет, да, Ашраф есть, да. Вот, наверное, твоя группа только и зашла, остальные меня закидывают. Пусть он напишет, Мне в следующий раз, наверное, там есть вариант копировать ссылку? Я ее буду копировать и рассылать, что ли? Да? Давай. Ладно, пока-пока. Ага, пока. Угу. Нет, это биостатистика, микробиологии тут нет. Я на портале, education портал. Дурацкие ссылки.
لا.
Тебя нет. So I should start the lecture. Do you hear me? Good afternoon.
Good afternoon, yes.
It is the constant link for the lecture, as usual, once 2 weeks. The leader should know this link. I am not going to send the link every time, but if you need, I will send you every time, but it is the constant conference. And I did ask the leader of the third group, 5103 group, to inform all leaders. You know this link, Ashraf, did you write
Yes,
it? Did you send them?
yes,
All
yes.
leaders?
I sent to all readers. I sent them the link of the lecture. Yes.
Right now?
Not right now. Maybe 5 or 6 minutes ago.
No, okay, no, it means today. But really, Pavel did send you this link 2 weeks ago, 3 weeks ago, and he told that you should share this information.
Yeah, I shared it, and actually, it's the same link that we have in our schedule. It's the same link.
So my leader from my group, they didn't find in the schedule. So and leaders are asking where the schedule is, where the link is. Could you explain them, please, Ashraf?
It's in the schedule. I have the schedule for all groups, for group 3, 4, 5, 6, and 7.
Okay,
And now I
okay.
schedule that this link is present.
Have you are you communicate with the leaders of another group?
Yes, I'm communicating.
So could you
the
send
link anyway? They have just to click and to share.
Could you send them the link and explain them that it is constant Zoom conference, and this link is used once a 2 weeks for Tuesday lecture? Okay.
Yeah, the link will not change. Yes.
No,
yes.
no. It's constant. Okay. So, and you have the lecture once a 2 weeks. So now it is 23rd of March, and then the next lecture will be on 6th of April. Not every Tuesday. Some students don't know, are confused. Okay. You have the lecture once a 2 weeks. So next lecture will be 6th of April. By the same Ashraf, could you explain them, please? Okay.
Sure, I will. Okay.
They are not able to understand, unfortunately. Okay, so the lecture is chemotherapeutic drugs and antibiotics, and very important information for pharmaceutical faculty students. Of course, this is there is a lot of information not possible to explain you in 40 minutes, and just main information, and then you will study on laboratory classes. So chemotherapeutic drugs and antibiotics. Now we are talking about treatment of infectious diseases, and we are not talking about talking about total treatment, because there are many preparations. We are talking about just preparation against microbes. Because in case of infectious disease, the main task is to eliminate the causative agent, or at least to stop the multiplication, but it is better to eliminate, of course. And different preparations are used, the antimicrobial, and chemotherapy is a specific treatment, and preparation, those are used, chemical, those are used, have different antimicrobial spectrum, because you know that different microbes are able to cause infectious diseases. Antimicrobial spectrum, antibacterial, and it is divided on gram-positive, gram-negative. So antiviral preparations, antifungal, and antiprotozoal, they are different, and not possible to use antibacterial preparations against viruses, for example. So for treatment of viral infections, they are absolutely special. So, and antimicrobial drugs against microbial pathogens have in general microbicidal or microbistatic effect. So microbicidal action leads to the death of microbes. It's better, of course. Microbistatic action inhibit the growth and reproduction. In presence of this preparation, microbes are not able to reproduce, but when the drug is removed, microbes restore viability. They're still alive. Anyway, both preparations are used because we should realize that many preparations are toxic for human beings, and it is possible to kill microbes by using some toxic substances, but at the same time, we will destroy many tissues and cells in human's body. So every year, chemicals suggest many new chemicals that are toxic for microbes. Many thousands of drugs are suggested by chemists, but just a few preparations finally will be used for treatment of patients. Okay, because we are going to get to use preparations with the very mild toxic properties. Okay, and in general, chemotherapeutic agents are divided into two groups: antibiotics and synthetic antimicrobial chemotherapeutic drugs. They are a little bit different because of antibiotics are produced by living cells. There are substances, those are produced by living cells, such as fungi, like Penicillium genus, for example, such as bacteria. In animals' body, antibiotics are produced in human body. So synthetic antimicrobial drugs are just produced by chemists, okay? They are artificial preparations. Now, some antibiotics are produced by a chemical way, but anyway, they have natural analog, okay? Because now we know structure of every natural antibiotics, and it is possible to produce by chemical way some antibiotics. Antibiotics, what are they? Chemicals of biological origin selectively inhibit reproduction or kill microorganisms. Uh, not all microorganisms, of course, antibiotics are able to act just on living cells. So they are able to inhibit reproduction or kill microorganisms that are really organisms, such as bacteria and fungi. Even protozoa are very often resistant to antibiotics, and we have to use chemicals. So you can memorize from now, viruses have cell structure. So viruses have natural resistance to antibiotics because they are not cells and there is no target of action for antibiotic. There is no any metabolism in virus particle. So antibiotics are not used for treatment of viral infections, never, just chemicals. Antibiotic classification. They are classified, number one, by origin. Antibiotic received from fungi, like benzyl penicillin, for example, from actinomycetes, streptomycin, for example, from polymyxin bacteria, then animal origin, and the plant origin, for example, chlorophyll. Very good preparation. Staphylococcus sensitive to chlorophyll. So then, number two, antibiotics are classified according to the method of obtaining. Maybe biosynthetic, natural, such as benzyl penicillin and chloramphenicol. They are naturally produced by fungi of Penicillium genus. Semi-synthetic, a little bit modified structure, chemical structure, ampicillin, for example. Then synthetic. Synthetic antibiotics are produced now by chemical way, but they are analog of natural antibiotics, like chloramphenicol, and is natural analog, and the artificial is levomycetin. Number three, antibiotics are classified in accordance with the mechanism of action. For every antibiotic, there is certain target of action inside the bacterial cell. And for example, antibiotics are able to suppress various processes in bacterial cell. Number one, some antibiotics are able to suppress synthesis of the cell wall components, exactly peptidoglycan. And what are they? Penicillins and cephalosporins. There are groups named, they are classified by chemical structure on groups. So penicillins group, cephalosporins group. It is possible to use Latin name of antibiotics and in English. But I guess for pharmaceutical faculty students, it is better to memorize Latin names. Anyway, you will study in Latin. Okay, so, and some antibiotics are able to destroy functions of the cytoplasmic membrane. Polyens and polymyxins, they act exactly on fungi cell. They destroy sterols. Those are in fungi cells. Fungi cells are eukaryotic cells. They have ergosterols. So, and the bacteria have no ergosterol, so they are resistant to polyens and polymyxins. No target of action. Then some antibiotics are able to inhibit the protein synthesis inside the cell, such as streptomycin, tetracyclines, erythromycin. And some antibiotics are able to inhibit the nucleic acid synthesis. For example, rifampicin, very modern preparation with broad spectrum of action. And then number four, antibiotics are classified in accordance with chemical structure. Beta-lactams, penicillins and cephalosporins, they have beta-lactam ring in the in chemical structure. So microlids and other lids like erythromycin, oleandomycin aminoglycosides, streptomycin, streptomycin, gentamicin in Latin, erythromycin, alendamycin, streptomycin, gentamicin, and tetracyclines group includes oxytetracycline and doxycycline. Polyions includes nystatin, amphotericin B, anzamycins include rifampicin and rifamin, and additional class includes chloramphenicol and griseofulvin, additional class. So then there is special group for treatment of syphilis. They are named anti-syphilitic drugs, and benzyl penicillins are used very often. Fortunately, treponema are sensitive to benzyl penicillins during many decades. Anyway, they did not develop resistance, fortunately. Penicillins, if patient has allergic reaction to these preparations, it is possible to use tetracyclines or maybe bismuth preparations, okay? And then the special group anti-tuberculosis drugs like rifampicin, streptomycin, kanamycin, there are antibiotics and there are chemicals, isoniazid and ethambutol. Then, unfortunately, during many decades of using of antibiotics, microbes did develop resistance to drugs that are used. And mechanism of antimicrobial resistance, number one, natural. Maybe they are resistant to some preparations by their nature. And number two, acquired. So mechanism of natural resistance. Number one, the absence of targets on the cell wall. For example, mycoplasma are resistant to penicillin action. Now you should realize why, because of mycoplasma have no other cell wall. So no target of action for penicillins and cephalosporins. So they are mycoplasma for treatment of disease caused by mycoplasma, penicillins and cephalosporins are not used. No effect. Then the impermeability of the cell wall for certain drugs. Some drugs are not able to go inside the cell. Then bacteria are able to produce inactivating enzymes, and then those are able to destroy antibiotics. And natural resistance, I told you, bacteria have no ergosterols, so they are resistant to polyen antibiotics. Viruses have no any metabolism, so they are resistant to antibiotics, all antibiotics. And then, unfortunately, bacteria and fungi, now it is proved, are able to acquire resistance, artificial resistance, acquired resistance. How it is possible? Changes in the metabolic activity of target cells. The number of targets for drugs may be decreased. Then genome changes may be due to mutations. This reaction that was the target now is not used in bacterial life. And unfortunately, there is very important reason, plasmids. And plasmids are small molecules of DNA. You will study next laboratory this week class, of course. Plasmids, they are small molecules of DNA, and they are located additional. So bacterial cells have nucleoid, the big one, molecule of DNA, and all of life important genes are in nucleoid. But there are some additional small molecules of DNA. They are named plasmids. They consist of genes, and information is encoded in these genes about some properties. And if a bacterial cell has additional plasmid, this bacterial cell has some properties. And plasmids are different. They differ by content of genes. You will know later, and they are named. They have names. So one plasmid is named resistance plasmid, in short R plasmid, and it consists of many genes where information is encoded about enzymes production. Those are able to destroy antibiotics. So in spite of bacterial cell has target for action, if the bacteria will produce enzymes for destruction of antibiotics, now antibiotics is destroyed. And everything is okay with this cell. Unfortunately, plasmid is transmitted from one cell to another. You will study this week genetics, how it is possible, even not just between bacterial cells, those belong to one species, between the bacterial cells of different species, and bacteria are able now to transmit our plasmid to fungi. No fungi did develop resistance to many preparations. So microbes are very adaptable. We live in microbes world, and it is the war between people and microbes, and we are not going to win in this war, I guess. Okay, so ways to overcome the resistance of microbes to chemotherapy. Of course, it's very big problem in medicine now, because of now many strains of microbes did develop resistance and not possible to use preparations. And what should we do? Number one, finding new chemotherapeutic drugs with a different mechanism of action. Chemical modification of non-antibiotics with protected active groups. Rational antibiotic therapy, it is prohibited now. World Health Organization did declare that it is prohibited to use antibiotics for prevention of diseases. Just if patient has clinical symptoms of diseases, it is possible to use antibiotic. And then the combined, do not disturb me, please. Turn your microphone off, please. The combined use of drugs. The combined use of drugs with various mechanisms of action, then the use of drugs that affect the early stages of reproduction, and the combined use of antibiotics and substances that destroy bacterial enzymes. Like, for example, antibiotic that is named amoxiclav. Amoxiclav consists of amoxicillin antibiotic and clavulanic acid. So amoxicillin is antibiotic, clavulanic acid is substance that is able to destroy bacterial enzymes. Now, different measures are used. And what else? Now, unfortunately, bacteria did develop resistance to many antibiotics. And now we have to treat every patient personally. Every patient should be treatment, we have to use preparations personally for everyone. Gonococci, for example, causative agents of gonorrhea, are differ by resistance to antibiotics, and Staphylococcus, Shigella, Gonococci, and tubercular bacteria, they did develop resistance. So we should isolate the pure culture, you know, how it is possible to isolate pure culture from every patient. And then we should do antibiotic sensitivity tests. They are named antibiotic sensitivity test. I don't like this name. Really, what does it mean? It means detection of the sensitivity of microbes to antibiotics. But in textbook, they are named like antibiotic sensitivity test. Really, we should do detect sensitivity of microbes. So after pure culture isolation, we should, there are many tests are used, and then the criteria for the activity of drug are the minimum inhibitory concentration, MIC, the lowest concentration of drug inhibiting the growth of the test culture. So, and we should measure this MIC by dilution test, and then it is possible to compare MIC of different preparations, and we have to use the preparation with the lowest concentration, with the lowest MIC. Then on laboratory classes you will study, and I hope you will understand what does it mean. What methods are used? Serial dilutions in liquid media, serious dilutions in solid media, then identification of methicillin-resistant Staphylococcus, and diffusion method. E-test is used, the Alomar method is used, there are many methods, and like modern method. Automatic systems for recording the result of serial dilution method, Alomar system, E-test, all of them are described in textbooks. Some of them you will study on laboratory classes. So, and antimicrobial chemotherapy complications. The toxic effect of drugs on kidney, central nervous system, inhibition of blood formation, dysbiosis, immunosuppressive effect, and allergic effect. So all preparations really have toxic effect, and we see side complications. So it is the end of lecture. Have you any questions?
Yes, please. What is the real difference between chemotherapy and antibiotics?
Ashraf, could you repeat your question, please?
Yes, the difference between chemotherapy and antibiotics.
I told you. No, now it is a common word. Chemotherapy, chemotherapeutic drugs, really. But in past, they were differ, absolutely. And I told you why. Because of antibiotics have natural origin. Antibiotics are produced by living organisms, different organisms, by microscopical fungi, by bacteria in animals' body, in human body. So antibiotics are natural biological products. Do you understand me?
Yeah.
Chemicals are
Can we say
produced.
that?
Chemicals are produced by chemicals. They are artificial substances. Now, some antibiotics are produced by chemical way because it is easier than isolate from producers. Okay. Anyway, every antibiotic has natural origin. Now, the common term is used chemotherapeutic. Okay.
Yes, yes, thank you. Yes.
But as for me, it's important. Maybe anyway. Every preparation has toxic effect. Every preparation, and before now by using of the first antibiotics was produced in USA at 1940, and it was penicillin. Penicillin was produced. It was isolated from Penicillium genus fungi, microscopical fungi, and really it was a miracle preparation. And it was time of World War II, if you know that. And many soldiers had a wound, and they really will, they the lethal outcome will be developed, but by using of antibiotics, and this penicillin did save many lives of people. And in time of war, it was used just for soldiers. And then after penicillin, many antibiotics were produced, and really doctors were happy. Doctors were happy. We have miracles preparation for treatment, but then they realized that many side effects are developed, and many organs are destroyed, and many organs. And sometimes patients are not here well. Sometimes a kidney are destroyed, absolutely so. And sometimes after treatment of any bacterial infection, like maybe patient has bacterial pneumonia, and the patient is going to die. Of course, it is necessarily to use antibiotic with broad spectrum of action for any case. It's not written. Antibiotics are differ by spectrum of action, narrow spectrum of action and broad spectrum of action. And of course, at hospitals, patients are admitted with very severe clinical case, and of course, to save their life, doctors did use antibiotics with broad spectrum of action. Means many bacteria are sensitive, including normal microbial flora. Of course, there is no antibiotic that is able to destroy just the one causative agent, one species, of course, no. If all gram-positive bacteria are sensitive, all gram-positive bacteria finally will be destroyed by using of this antibiotic. So, and if antibacterial antibiotic is used, and really it should be used to save the life of patient. And finally, the patient did recover of bacterial pneumonia, but finally, very often children, finally this patient did die. And doctor did realize that this patient finally did die of candidiasis. Candidiasis caused by own microbial flora, fungi from Candida genus. Candida genus, you didn't study yet, but you will study. I gave you the lecture, normal microbial flora, by the way. Okay, so, and Candida are able to exist in very small amount in human body, and they are named normal microbial flora, but in very small amount, just a few cells should be detected by using of microscope. And multiplication of these fungi is suppressed by bacteria, of course. There are very complicated relationships in microbiotes in human body. And but by using of antibiotic with broad spectrum of action, the amount of bacteria will be decreased. All bacteria and Candida has opportunity to multiply now, and they will multiply a lot. And finally, generalized candidiasis was developed. It's possible to be developed, and patient finally will die of candidiasis. Okay, so doctor did realize that there are many side effects, and and we had to use antibiotics after pure culture is isolated, because every patient should be treated personally. In some cases, like in case of syphilis, okay, treponema are sensitive to penicillin till now. And we are not going to isolate pure culture. Anyway, cultural method is not used in case of syphilis, because they don't grow well. It is very difficult. Without any isolation of pure culture, a diagnosis proved by detection of antibodies for treponemas in the serum or blood, and by PCR, polymerase chain reaction, and the treatment is done by using of penicillin. If bicillin is used now, but if patient has allergic reaction, sometimes allergic reactions are developed to antibiotics. We have to find another one. In case of cholera, for example, cultural method is used, but we are not going to use the test for detection of sensitivity of bacteria to antibiotics. We know that Vibrio cholerae is sensitive to tetracyclines groups antibiotics, and it is possible to use any one. Doxycycline is the modern antibiotic from this group, doxycycline. So sometimes not necessarily to isolate pure culture and to do antibiotic sensitivity test. But in case of staphylococcus, in case of Shigella, in case of tuberculosis, it is necessarily, okay? We should isolate pure culture and we should realize what antibiotic should be used. So any questions? Have you any questions?
Can you say that bacteriophages are antibiotics?
Oh, no. Oh, good question. Bacteriophages are viruses or bacteria. Viruses, they are viruses. But the bacteriophages are able to reproduce in bacterial cells only. And they are able to destroy bacterial cell after
Because they are
reproduction.
natural, like
They are natural, and
natural.
they are not dangerous for human beings. They are not able, bacteriophages are not able to reproduce in human beings' cells. They are able to reproduce in bacterial cells only. So they were used before antibiotics. Bacteriophages were used, they were produced, and they were used for treatment of bacterial infections. And they have species, we will study bacteriophages this week. Bacteriophages have species specificity. They are able to reproduce just in 13 species of bacteria, genus specificity, and they were used very widely. They have no side effects. But then antibiotics were detected, and doctors did forget about bacteriophages and they did believe in antibiotics. Now again, doctor did realize that we have many side effects and complications due to antibiotics treatment. And in Russia, for example, now a lot of antibiotics are produced for treatment of bacterial infections. Of course, if patient is going to die and the patient is admitted at hospital, of course, antibiotics are used. Many preparations are used, antibiotics including, of course. Okay.
Thank you.
Oh, welcome. Any questions? So the next lecture will be in 2 weeks, and please, the leaders try to get the link. I saw that you have this link, and the next lecture will be on 6th of April, okay? In 2 weeks from now. So
Okay.
goodbye.
Goodbye.
Doctor, we have any task to complete on the portal?
No, test there. You should answer test on portal. Thank you so much. Now go on education portal and answer the test for lecture. And I will check attendance of lecture by answers for test, okay? Informal student, please.
Okay.
The leaders, you are responsible for students. Okay, inform them they should answer test on educational portal. If student did not answer, student did not attend the lecture. They are open, tests are open till the evening, so do it today.
Okay.
Then do not be surprised.
It's test for fourth lecture. The fourth lecture.
Test for